If you destroy 75% of a human liver, the remaining tissue will rapidly expand, stitching itself back together until the organ is whole again. It is a dense 1.5 kilogram structure tucked securely beneath the right side of your diaphragm. Biological regeneration on this scale requires massive amounts of energy. So why did evolution equip this specific organ with such an extreme survival mechanism? The liver requires this ability because it operates on the front lines of a constant crisis, sustaining regular damage to keep the rest of the body alive. The danger begins with digestion. When you eat, your intestines absorb the nutrients your cells need, but they also pull in useless byproducts, raw fats, and concentrated toxins. If this raw, unfiltered blood from your gut were allowed to circulate directly to your brain or your heart, the toxic load would be lethal. To survive, the human body relies on a constant 24/7 inline filter, a biological bottleneck that sorts threats from nutrients before entering general circulation. This diagram shows the two distinct circulatory routes converging to force blood into the liver. The first is the hepatic portal vein, delivering 80% of the blood supply straight from the intestines. This flow is packed with nutrients and toxins. The second is the hepatic artery. This delivers the remaining 20% of the blood supply directly from the heart, providing a concentrated payload of oxygen. This dual delivery system creates the necessary staging ground. The intestines supply the raw materials to be processed, while the heart supplies the oxygen fuel required to run the machinery. Inside the organ, blood enters a gauntlet of over 1 million hexagonal processing units called hepatic globules. As it enters, sinusoidal endothelial cells instantly intercept and neutralize raw toxins. Next, hepatocyte cells act as manufacturers, synthesizing crucial proteins and transforming raw carbohydrates into energy. Simultaneously, hepatic stellate cells act as pantry workers, storing fats and vitamins. Finally, Kupffer cells act as the recyclers, consuming dead red blood cells and converting the remaining toxic waste into a new byproduct. This microscopic hexagonal gauntlet divides the labor, managing hundreds of distinct chemical conversions simultaneously as the blood moves toward the central hub. Once processing is complete, the cells convert the toxic hazards into a bitter green liquid called bile, which is funneled into the gallbladder. This bile trickles into the intestines, where it breaks down fats and neutralizes extra stomach acid. Meanwhile, the cleaned and detoxified blood flows out through the central vein of the lobule and travels upward to connect to the heart. Only after passing through this biochemical stress test is the blood deemed safe enough to be distributed to the rest of the body. Because the entire body relies on this constant filtration, any disruption to the factory line triggers a rapid, systemic cascade of failure. In the case of hepatitis, viral inflammation corrupts the tissue, effectively shutting down the cellular production line. Jaundice is another mechanical failure. When dead red blood cells aren't cleared out by the recyclers, a yellow pigment called bilirubin floods the bloodstream and discolors the body. These conditions are not random. They are the direct physical consequence of specific cellular workstations failing to do their jobs. The liver is characterized by its resilience. It can recover from massive physical trauma, yet remains vulnerable to chronic, long-term toxic overload. The organ maintains a state of constant coordination, running constant filtration, complex chemical synthesis, and nutritional storage in unison every single second of our lives. We have to actively protect this biological bottleneck. If your kidneys fail, there is dialysis. But if this central processing plant stops, there is no artificial machine that can replace it.